Hello, everybody. Welcome back. We are going to continue our discussion of sexual reproduction from Chapter 10 with the meiosis topics and expanding it into inheritance patterns. So this is kind of what happens after those gametes are produced. So in Chapter 10, we talked about the steps taken in the gonads, the ovaries and testes, to produce the haploid egg and sperm cells. So now what we're going to do is take a look at what kind of dictates, what laws of inheritance determine what happens when the egg and sperm meet, and what are the probabilities of some of those traits carried on the genes, carried on the chromosomes. What are they going to look like in the offspring? So that's kind of our goal in this chapter, is to kind of just apply meiosis to real world examples of genetic inheritance. So let's get started. All right, so before we get into the nitty gritty, if you will, or the rules that govern the traits we inherit, we got to talk a little bit about what inheritance is. It's, in general, the basic idea is it's the physical characteristics, the biological characteristics from one generation being passed on to the next generation. And what we know now about this is that that information is encoded in genes. So remember, genes are sequences on the DNA that code for a particular outcome, maybe a protein or a process in the cell that gives that organism some particular characteristic. I put up a picture of the Baldwin brothers here so you can see some resemblance. They're not identical. They have the same parents. So they have some similar characteristics, but they, just like any other set of siblings outside of identical twins, they're going to have some similarities, but some uniqueness as well. And remember, that's going to be due to those events in meiosis, right? The crossing over in prophase 1, the independent assortment of metaphase 1, and the random fertilization of which sperm is meeting which egg. So each one of those combinations will give you a unique offspring. All right, so let's take a look at genes, kind of review this a little bit. So remember, we have chromosomes, or long individual pieces of DNA made up of A's, T's, C's, and G's. So a certain region on a chromosome that has a specific sequence of those letters, that is what we call a gene. And that gene codes for a particular characteristic. And there's different forms of those characteristics. I think we've seen the word allele before. So also the physical location of where that gene is on the chromosome is called the locus. Locus is singular. You might see it loci. L-O-C-I is the plural form when you're talking about more than one locus. And so the genes found on these chromosomes, remember, when we're talking about diploid cells, right? Diploid cells have 2 copies of each chromosome. You're going to have 2 copies of that gene. Sometimes the genes can be the same. Sometimes they can be slightly different. That allele form, the allele is the variation or the option that that gene could take. So over here in the picture, that first pink bar, if you will, on this pair of homologous chromosomes, it's the same gene, right? Because it's in the same location. And because they're showing it as the same color, they're saying it's going to be the same form of that gene. So it's going to be the same allele. The second band on that picture is the purple bar, and so this is also showing it the same color. It's the same gene because it's at the same locus, and they're showing it as the same color, which is representing it's going to be the same allele form of that particular gene. And then the last one, the third gene that they're showing, the green band, it's the same gene because it's the same locus, but now they're showing one is a dark color and one is a light color. They're representing that as different alleles for that one particular gene, so different forms of that gene. So we could do taco tongue or not taco tongue, widow's peak or no widow's peak, attached earlobes or no attached earlobes, curly hair, straight hair, albino pigmentation, normal pigmentation. So those are examples of some alleles that could be found on your homologous chromosomes. All right, so here is, we're going to introduce a couple of new words that are very important when we take a look at genetic problems and kind of solving this probability of what the offsprings are going to be. So when we're taking a look at those alleles, again, 2 different versions of the same gene, we have some different combinations that could potentially show up on those homologous chromosomes. So if we take a look at, let me get my pen going here. If we take a look at this first pair, so let's say this chromosome is from mom and this chromosome is from dad, Okay, so these were the gametes that were made in meiosis. So we've gone through the process of meiosis. We have haploid gametes. So chromosome number 1 is in the egg, chromosome number 2 is in the sperm. They've come together, they've fertilized, they've fused, and now we have our diploid daughter cell that has now 2 copies of every chromosome. So this is kind of that developmental stage at where we're at. This is post-fertilization. We are back to a diploid cell, and 1 copy came from mom and 1 copy came from dad. So that's kind of these blue chromosomes that we're going to be taking a look at. So in this first option, maybe mom and dad both were carrying this, what's called the dominant allele. We'll dive into dominant recessive in a little bit. We're just denoting that as the capital letter B. So that gene B is at that particular locus, and it has this capital B form. Okay, so mom in her egg, whenever she did her meiosis, she happened to donate this chromosome that had the capital B. And then dad, and when he made his sperm, he donated also the same chromosome, and his form of that allele also is capital B. So this diploid daughter cell, this now new organism, it is going to, those are the only options it has. It has 2 capital B forms, these alleles for that particular gene. And so when you have 2 of the same form of allele, we call this homozygous. Okay, so homo meaning the same, zygous has to do with alleles or chromosome form or trait form. So when you are homozygous for a particular trait, that means in all of your cells that are diploid, right? Diploid cells, these are all of our body cells. If you're homozygous, you have 2 of the same form of that allele. In this case, we have 2 forms that are both the capital letter. Could we have another possibility for homozygous? Well, yes, we can. Let's take a look over here. Let's say this came from mom and this came from dad. Okay. Now in this example, mom donated her chromosome that had the little b, the lowercase b. So this is what we're going to call recessive. We'll define that, like I said, in a little bit. So mom is donating the little b, and dad happened to donate the little b on with his sperm. The nuclei fuse. Here you have your diploid daughter cell, and now this organism, the only form of this allele or this gene that they can express is this lowercase form, the recessive form. So this organism is also considered homozygous for this particular trait. So when we're taught, how do we differentiate between these 2 forms of homozygous? So we'll just say homozygous dominant for this one, okay, because it's a big B, capital letters are used to denote dominance. And then over here we have homozygous recessive is what we can say. So that's the little b, so 2 recessive alleles. So you could be homozygous dominant, or you could be homozygous recessive, okay? But there's some options in the middle here. What if mom, right? So let's use this set over here. So what if mom was able to donate a dominant allele and dad donated a recessive allele? Okay, so now this diploid offspring, the child of this mating, is going to have 1 capital letter and 1 lowercase letter, or 1 dominant allele and 1 recessive allele. So when you have 2 different forms of the allele, this is what we call heterozygous, because hetero means different, right? Homo means the same, hetero means different. So heterozygous are organisms that have 1 of each allele for that particular trait. Now it's showing it twice, but it's both 1 capital letter, 1 lowercase letter. The reason why they're showing it twice is because in this first heterozygous pair, mom donated the dominant allele. In this second heterozygous pair, dad donated the dominant allele. So it could go either way. So you could get a dominant or recessive from mom, a dominant or recessive from dad. And then when we look at the outcome, what's this combination of letters? If it's both the same letter, we call that homozygous. If it's 1 of each, we call that heterozygous. So I'm going to be using those terms a lot as we progress through this chapter. So if you're still kind of struggling with that, maybe you can pause the video, go watch an animation or reread that section in the textbook, because you need to have a good grasp of these terms as we progress through this chapter. You also might see heterozygous be called a hybrid because it's got 1 of each. It's not a purebred, it is a hybrid. Okay, so a little bit of a history lesson. So this was taking place late 1800s. So I know we're kind of in the almost 2020s. So a little bit more than 100 years ago, we really didn't know about genes or alleles or chromosomes, this idea of molecular inheritance. People knew for hundreds of years about traits, because there was artificial breeding of like dogs and farm animals, cows and sheep and plants, looking for people realize the traits that they wanted. If they wanted to enhance those traits, then they would have to cross or mate the organisms that had those traits. And then the offspring, you would have a higher chance you would get the offspring of that mating to have that particular trait. So that was known. But they didn't know why. They didn't know that it was chromosomes. They didn't know it was genes. They didn't know there was these things called alleles. They knew there were different forms, because you might have, if you're trying to get a white poodle, there's brown poodles and black poodles. And so they knew there was variations, but they didn't know the scientific bits behind it. So in the late 1800s, about 1880 something, this guy named Gregor Mendel, who was a monk in what's now called the Czech Republic, he worked with plants. And so he is considered the father of genetics, because he came up with some of the principles that we still use today, which is amazing because he didn't know anything about chromosomes and meiosis at that. Meiosis was just getting discovered about the time he was doing his experiments. So there's kind of a drawing, a painting of him working with his pea plants. He studied the garden pea. You might have actually some of this growing in your backyard. You may have seen it. Sometimes it grows along fences on the sides of roads. It's a very common plant. He studied math and botany at the university level before he became a monk. So he had kind of an education before he went into that field. And so he had a garden, and he was just making these observations. And he was very good at keeping his track of his data. He did good scientific experiments, only tracking one variable at a time. So he didn't get mixed up with, could this be the thing? Could this be the thing? So he usually just followed one trait at a time. We'll see he did actually expand to follow two traits to reinforce some of his observations. So there's him working with his garden peas, and I just found a picture of a garden pea, just kind of a Google image to show you. You've probably seen or eat these things before. They're pretty tasty. All right, so Mendel was actually kind of lucky by picking the garden pea as a plant to study because it had some really good characteristics. If he would have picked, an animal model or some other plant model that had kind of weird genetic inheritance patterns, he may not have come up with the rules that he did. So thankfully, luckily, right, sometimes science is just good luck, is that he picked a plant that followed these pretty strict inheritance patterns, which set up the rules for almost everything else to follow along. So the flowers that are made by these pea plants, they have distinct male and female parts. So there's male structures that produce the pollen. So in plants, pollen would be like the sperm. So that's going to the male gamete. And there's pretty clear female parts that make the eggs, right? So we have sperm and eggs. They are the end results of meiosis, just like we saw back in chapter 10. The way that the flower is structured is that once fertilization happens, the flower petals kind of fold around those reproductive parts, and so you minimize accidental cross-pollination. So you don't get more than one sperm cell fertilizing the eggs, because that would kind of confuse the results. Another interesting fact about these pea plants is they can self-fertilize. So that usually isn't a good idea in the animal kingdom. So that would be what we call an incestuous relationships or close related matings. So that results in some pretty severe problems in the animal kingdom. But in plants, it's not that big of a deal. So you have a lot of plants that can self-pollinate or self-fertilize. So that allowed him to be able to keep these particular traits running through a plant kind of pedigree, if you will. And he also noticed that for whenever a plant was producing this particular trait, if it was always producing that trait, it always produced that trait. So he called these true breeding. So my head's kind of cutting off this word there, but plants who are homozygous, which he didn't know that word then, but we're going to use it. Plants who are homozygous, which means if they had purple flowers, they always bred purple flowers generation after generation. If you did this self-fertilization, you're not going to sneak in another color somewhere. So he established these true breeding plant lines. So whatever trait he was looking at, he was able to keep them just having that trait with this self-fertilization ability. So here's kind of a drawing picture of the flower. And so there's an intact pea flower. And then what he was able to do is carefully dissect out those reproductive parts. So then he could manipulate where the when and where the pollen was going to go. So if you wanted to self-fertilize, he would just take some of the pollen from the stamen, which is the male reproductive part, with like a little paintbrush, like a little brush, and then he would transfer that pollen to the carpel, which is the female part. And so this was a self-fertilization example, or if he wanted to do a cross mating, he would just take he would cut all of the stamens off, right? He would just cut them all off. And then maybe there's a stamen of a neighboring flower that he wanted to transfer over to the carpel. And so that allowed him to cross pollinate from one different plant to another plant. So that was what he was able to do. He could self-fertilize and he could also cross-pollinate from different plants. And then he would just kind of tuck everything back up into those folded flower petals, and then it would prevent pollination from something else that he wasn't specifically looking for. All right, so he actually studied 7 traits. So the main one that we're going to start with is flower color. So this one right here in the middle. So these garden pea plants come in 2 options. We now call those alleles, right? So the gene is flower color. So that's the specific sequence of DNA at that particular locus on the chromosome, and it has 2 options. It will either give the instructions for purple flowers, or it will give the instructions for white flowers. So this is kind of a really obvious expression of that particular trait. He also took a look at some characteristics for the seeds. The shape, they're calling it form here. So the seed shape could be round seed or kind of wrinkly. The cotyledon is just part of another word for seed. So the seed color, it could be yellow or green. So you could have yellow pea seeds or you could have green pea seeds. The flower color, purple or white. The pod, the shape of the pod could either be kind of puffed up and full, or it could be constricted like it was put in a vacuum pack or something. We could have different colors of the pods. So you could have pea pods that were green and pea pods that were yellow. You could have where the flowers are located on the stem. So if they were axial, they were kind of coming off at different locations on the stem. So you could have a flower here and a flower here and a flower here. Or you could have the flowers all coming off. I know my head's blocking this. Terminal means all of the flowers are coming off of the very top part of the stem. So they could kind of come up along the axis, or they could all come off the top. So that's the location of the flower. And then the last one was how tall. So you could have tall plants and you could have short plants. So that's what this is right here. Behind my head, it will say dwarf or short. So he actually followed 7 different traits within these plants repeatedly and a lot. So we're talking like thousands of plants. He didn't just plant like 3 of each kind and try to follow those traits. He did lots of repetition, which we know is a good part of scientific method. And so he was able to be confident in his results. He was seeing these same patterns over and over and over again. All right, so we got a little bit more, some new vocabulary to add on to our list. So we have allele, which is the different forms of the gene. We have homozygous and heterozygous, which are homozygous, you can have 2 of the same alleles in your diploid organism. Heterozygous, you have 1 of each, 1 dominant and 1 recessive. So now when we're going to get into these crosses, right? So we have parents, we have offspring, we got to keep track of those things. So these are the terms that Mendel came up with. So when you have your first kind of generation, these are what we call the P generation or the parental generation. This is the first kind of set of your genes that you're starting to track. When the parents mate and they produce their offspring, this is what we call the F1 generation or filial. First filial is kind of like an older term that means offspring. So it's the first filial generation, we call that F1. And then when F1 can self-fertilize or 2 different F1 generations cross, we just call this F2. And then you can continue that on. So he did multiple filial generations. We're really only going to see the parental F1 and F2. We don't need to go beyond that. And so here in the diagram, we have a what's called true breeding purple plant. So this one right here. So true breeding means it's homozygous dominant in this case because purple was the dominant trait. So this would be like big P, big P. I can put those here. Big P, big P. And then he fertilized that with a true breeding white, which means homozygous recessive. So this would be the little p, oops, little p. So that was your parental generation. So he crossed big P with little p, right? And so then the F1 generation, if you took a look at it, all the plants were purple. It's like, well, wait a minute. So that was one of his first kind of big question marks in his observations is I had a white plant mate with a purple plant. How come 100% of the offspring are all purple? That doesn't make sense. But he didn't give up. So he's like, well, let me try something else. What if I were to self-fertilize that F1 generation, get all those purple plants of this F1 offspring mate with themselves? Okay, so that's what he's going to do. So this, I just put the same picture up there so we can do, I asked that question a little bit too early. So our true breeding purple plant, true breeding white gets you 100% F1 generation. So here's that question, what happened to the white flower trait? Well, here it is. So this was the 100% purple, and then he self-fertilized, so cross taking all of the pollen from that same flower to the female parts of that same flower. And what he noticed in the F2 generation was that white color came back out, but it wasn't in an equal proportion. He saw that about 75% of the flowers were purple, with only about 25% of the flowers were white. So it kind of skipped a generation. And sometimes you hear that, oh, you know, that trait skips a generation. Well, this is actually kind of where that came from, is that when you have those true breeding parents give you that hybrid, which we're going to be identifying as a heterozygous a little bit later, when you cross those heterozygous, that recessive trait's going to pop back out in that third generation, in that F2 second generation offspring. So we're going to be using this example to introduce a tool later on called the Punnett square. Okay. So with his traits, I'm bringing back all the pictures of his traits, he noticed whenever he did these crosses, the trait that showed up in that F1 generation, so if you had two true breeding parents, and the trait that showed up on the F1, this is what he called dominant. So it overshadowed and kind of hid the recessive allele until that F2 generation, that recessive allele came back out, but in a smaller percentage. So for these particular traits, I think on the top row, nope, it's not the top row. I think the round is dominant, and the yellow is dominant, and the purple is dominant, and the full is dominant. And I don't know about these ones. I don't know who's dominant in these ones, but it's not important for this class. But just know, with his experimenting, he was able to identify which traits or which alleles of each of these genes were dominant and which ones were recessive. But again, he didn't know it was genes. He didn't know it was chromosomes and meiosis was the process. He was just making observations and recording his data. All right. Okay, so we've got a couple new words, lots of new vocabulary with genetics topics here. So when we are trying to then represent this information about the organisms, we can describe the specific combination of those alleles, which would be like the letter combinations, but then we can also describe the physical appearance of what's happening with those traits. So we use the word genotype and phenotype. So when we are talking about the specific combination of alleles, the letter combinations, is it homozygous, is it heterozygous, homozygous dominant, homozygous recessive, we call this the genotype, okay? To contrast that, whenever we're talking about what is the expression, what's the physical characteristics of this, we call it a phenotype. So genotypes are the genes, phenotype is the physical characteristic, which is kind of nice because they both start with PH. That might help you. So let's take an example of this purple flower trait that we're taking a look at. So if we know that purple is dominant, right? So we can start using where these letters come in. So the letters are just a random assignment. Sometimes it's the first letter of the trait, or sometimes it's the first letter of the dominant allele. You can choose. It could be any letter in the alphabet that you want, but you only pick one letter. You don't pick a different letter. You don't go P for purple and W for white. It's one letter per gene, and you use the capital and the lowercase to represent the different alleles. So in this case, the capital P is going to represent the purple allele form, and the lowercase p is going to represent the white allele form. So you keep the same letter because we're going to do some double letters here in a little bit, so you don't want to get confused. So whenever you are setting up your problems, pick one letter for the gene, and then the different allele forms are going to be represented by the capital and lowercase letters. That will really help you when you're working through these problems. All right, so we've assigned a letter, P. P for flower color. We could have possible genotypes. So what are all the possible genetic combinations for a diploid organism with this one trait? Well, there's two options. So that's going to give us three, there's two options for the two alleles. So we have three different potential combinations. So here's our homozygous dominant, big P. Here's our homozygous recessive, little p, and here's our heterozygous, right? So those are all the options. That's not too bad. So whenever you are tracking one trait with two alleles, you're going to have three genotypic possibilities. 1, 2, 3. One trait, two alleles, three genotypes, okay? And then if we take a look at the phenotype, so phenotype is a physical expression. So we have to understand that whenever you have at least one of those dominant alleles, you will physically express that dominant phenotype, okay? So let's take a look at our homozygous dominant. So that's this guy right here. So if you have big P, you have two of those dominant alleles, you are going to be expressing the dominant phenotype. So it's going to be a purple flower. If you are heterozygous, you have at least one of the big P's, right? You still have a recessive, but remember recessive, its definition is that whenever it's with a dominant, the dominant overshadows. So even though that heterozygote organism has a recessive allele, because it also has a dominant allele, it is going to be the dominant phenotype. So in this case, a heterozygote will be a purple flower. And then for the homozygous recessive phenotype, because it doesn't have any capital letter, no dominant allele whatsoever, it has to show that recessive allele. So that's going to be a white flower color. So that's its phenotype. So we have two phenotypes, purple flowers or white flowers. We have three genotypes, homozygous dominant, heterozygous, homozygous recessive. Okay, so that's how these patterns are going to go. So again, I'm going to be using these terms a lot as we progress through the rest of this chapter. So you should be comfortable with homozygous dominant, heterozygous, homozygous recessive, genes, alleles, phenotype, and genotype. Okay, so this is the language of inheritance. Okay, so let's take a look at what's happening, trying to apply some of that meiosis onto this inheritance patterns. So when you have a homozygous parent, right? So this is your diploid parent, and these are your haploid daughter cells, right? So they're 1N. I'm going to do a lot of PowerPoint writing today, so bear with me. My mouse is not the best tool for writing on a screen. So we have diploid parents. Okay. Now this is going to be their stem cell, if you will. So go back at the very beginning of meiosis. You have a diploid parent cell, and it is going to go through your double rounds of meiosis, and by the time you get to the end, you have your haploid gametes, and that's exactly what's happening here. So this arrow is representing meiosis. So what we have is the purple parent, and they have egg or sperm, so it could be male or female. We're not going to really assign sex to our plants. But if it's a male plant, or it has both, right? What am I thinking? It's got male abilities to produce sperm and eggs. So in this parent plant, the diploid stem cell will eventually produce haploid daughter cells, the sperm here in the oval, and the egg is the circle. And take a look at what its options are. So if it is homozygous on its two homologous chromosomes, right? So you guys remember homologous chromosomes, they get pulled apart in metaphase 1 and the chromatids get pulled apart in anaphase 2, sorry, anaphases. So homologs get pulled apart in anaphase 1, chromatids get pulled apart in anaphase 2. The only option for this particular trait to end up in those gametes is the capital letter, because that's all it has, right? So your homozygous dominant parent can only produce egg and sperm that carry that option. So it's going to be the same thing for the white parent. So it's got recessive. So here we do our meiosis, and we get our haploid daughter cells that can only carry that recessive allele. Okay, so true breeding homozygous parents, their gametes can only produce or give to the next generation whatever allele they have, because they're homozygous. They don't have a choice. Okay. If you were to cross that, so right, so this was our P generation to our F1, so the F1 generation from our homozygous parents. So we could have gotten the capital P from the sperm and the lowercase p from the egg. So this would have been our true breeding. This is our parental haploid genes. So you could have had two options. The sperm could have been carrying the dominant allele and the eggs could have been carrying the recessive, or it could have been the sperm carrying the recessive and the eggs carrying the dominant. Either way, look at what you end up with. That F1 offspring, how would you say its genotype was? What's its combination of letters? Okay. So the F1 offspring is now heterozygous because it has one of each. And it doesn't matter whether it got the dominant from the mom or the dominant from the dad, you're still going to end up with the same genotype and the same phenotype, right? So if the genotype is heterozygous, the phenotype is going to be purple. And that's what we see. And so that was his kind of Mendel's first big question is like, how can I get 100% purple offspring when I had a white and a purple plant mate? Well, because that purple, all of the sperm or all of the eggs carried the dominant allele, and then the other gamete carried the recessive allele. So 100% of the offspring are going to have at least one of those dominant alleles, so they have to show that dominant phenotype. So then what did he do? He crossed F1 generation. So this is where we get a lot more options when we're taking a look at those heterozygote, right? So here's our heterozygote F1 plants that then he self-fertilized. And so you could have lots of options. So you could have the P's. There's these potential possibilities that both the sperm and the egg could have gotten the capital P. There's a possibility, so that would be this right here. There's a possibility that both the sperm and the egg get the recessive allele, okay? And then there's the possibility that the sperm could be purple and the egg could be white, or the sperm could be white and the egg could be purple, right? So hopefully you're not getting lost in all of these letters and abbreviations. So if we take a look at this, so then this was that third generation, that F2 offspring where that white allele came back up. You can see we had 3 out of 4, so 75% of the offspring right here. So this was the purple phenotype, right? So remember, phenotype is the physical expression. And we know that because we have a homozygous dominant phenotype, or sorry, genotype. We have heterozygote phenotypes. Sorry, I'm getting my words mixed up. Homozygous dominant genotype, heterozygous genotype, and then the white, because there's no dominant allele, that homozygous recessive, that's going to give you that 25% of the population. Now, doing it in pictures like this is kind of visually helpful, knowing that those eggs are coming from the haploid parents, but there's a better tool. So I think this is where we're going to introduce this Punnett square. Yes. So I like Punnett squares because they make all of this stuff we just talked about a little bit easier, kind of this bookkeeping, keeping track of all of these letters. So let's say we have this F1 generation. So this was our F1. This was the one that 100% of the offspring were purple after the cross of the white and the purple, right? So we have a true breeding purple mate with a true breeding white. 100% of their offspring was purple, and then he crossed those purple F1 plants with themselves, and so this is where he saw that 75 to 25 split. So when you have your diploid parent cell, right? So and it's heterozygous. So remember, this is diploid. Okay, because it has 2 copies. So this would be the homologous chromosome. So this is maybe this big P is on the mom's chromosome and this little P was on the dad's chromosome. So those are the homologs, and then they split in meiosis. And so by the time we get done, we have our haploid gametes are going to get one of each because they're haploid. So we have the possibility that the egg or the sperm could have either the dominant or the recessive allele. So that's what's displayed up here. So maybe the egg is on the top, so this is the female. And because she was heterozygote, right? So she was a heterozygote, she has the possibility of producing eggs that are purple and the possibility of producing eggs that are white. So we have to include all the possible combinations, okay? And if the sperm, in this case it was itself, if this was the sperm, there could be a possibility that half of the sperm or the pollen was carrying the dominant allele and half of the pollen carries a recessive allele. Okay, so again, it's all the possible combinations. So then you just fill in the Punnett square. So you bring in this one letter here, and then this one letter here, and what you end up with, this is like would be the fertilization event. So your egg met your sperm, the sperm brought in the capital P, the egg brought in the capital P, and now you have your diploid zygote that is now back to 2 copies, but because it got a dominant from mom and dominant from dad, we're going to say that's the homozygous dominant genotype with a purple flower phenotype. All right. So then if we take a look at the homozygous recessive, what if the egg donated a lowercase p here? Oops, I'm going to erase that. I'm going to go all the way down. So let's say the egg donated the recessive allele and the sperm donated the recessive allele. So then what we end up with is our homozygous recessive diploid daughter cell showing that white phenotype. Okay, so that's the 25% homozygous dominant, 25% homozygous recessive, but now we have to count for the rest. The rest of the 50% are these heterozygotes. But if you remember, or keeping track or kind of learning this pattern, heterozygotes are always going to show the dominant phenotype. So this is where we get the 3-to-1 ratio that Mendel came up with. And again, he did this thousands and thousands of times. He didn't just, cross these F1 generations and get 4 plants. He crossed these F1 generations and planted like 3,000 seeds, or we'll make the math easy, 4,000 seeds. And of the 4,000 seeds, about 75% of those, 3,000 of them came up purple, and about 1,000 of those came up white. Okay? But this is a possibility, it's a probability. So if you did a mating and you created a baby, there would be a 75% chance that the baby would have that dominant phenotype. That doesn't mean that, and if the baby or it had 25% chance of the recessive phenotype. So then let's say the family has another baby. Just because they had that one recessive phenotype baby doesn't mean they can't have it. It just means there's another 25% chance that it can show up again the next time that they mate based on their parental genetics. Okay, so I've come together, or I put together a list of steps when you're working through genetic problems. You might be seeing these coming up in the homework, you might be seeing the quizzes as you're practicing through questions at the end of the chapter, or maybe exam questions that are coming up. So it's a good idea to kind of approach these genetic problems, sorry, I kicked the desk, approach these genetic problems in a stepwise fashion. So once you get this pattern down, then it's a lot less likely that you're going to get lost amongst all the letters and this new terminology. Okay, so the first thing in your problem is assign a letter for the trait that you're following. 1 letter per trait, right? We're not doing doubles yet. So just 1 letter per trait, your choice. So for some examples, you could do C for hair color, E for eye color, H for hair texture, whatever you want it to be. I usually like to use letters where it's a clear distinction between the capital and the lowercase, like H's and E's and B's and D's. When you get into things like C's or S's, it's a little bit harder to tell the difference between the dominant and the recessive. You just have to draw it in a different size because the shapes of the letters are basically the same. Okay, second thing is make a genotype and phenotype key, kind of like a little clue to yourself what's going on. So that would be, let's do hair texture, because I'm going to use that as an example a little bit later. So if we're going to do a genotype key, remember genotypes are the letter combinations. So you could do homozygous dominant, you could do heterozygous, and you could do you could do homozygous recessive, right? So those are all your possible genotypes. So that would be your genotype key. Your phenotype key would be like, I think I have it printed here. So big H, homozygous dominant, and big H, heterozygous, that's going to be straight hair. So that's letting you know that straight hair is the dominant phenotype. And then little h, or homozygous recessive, that's going to be your curly hair. So if you have that set aside, so you could say this is going to be straight, this is going to be straight, and this is going to be curly. That's your key. So whenever you work through your problems, you're always going to go back and say, oh, what do I have? What's my genotype? What's my phenotype? And so you can kind of keep track of that. Okay, so then if you have the problems in the question, or the statements in the question might be giving you clues as to what the parents are, identify those genotypes, write them down, look at your key to make sure if it says the parents are homozygous dominant or heterozygous or homozygous recessive, you have your key just to go back and take a look and say, okay, so here's the letter combinations that represent that phenotype that's being expressed in the problem. So that might be like a heterozygous crossing with a homozygous recessive. So we'll see that on the next slide when we approach this problem. Okay, then you'll set up your Punnett square, just like a 4-box box. Fill in the boxes, right? So you're setting up your parental genotypes, one parent on the top, one parent on the side. You fill in your boxes, and then you analyze your offspring and answer whatever the question is asking. You might say, what are the genotypes of the offspring? What are the phenotypes of the offspring? What's the likelihood that they're going to have a homozygous recessive or a heterozygous? So then you can answer the questions based on what you have in your box. Okay, so let's go through this. So here would be a question. A woman who is heterozygous for straight hair mates with a man who has curly hair. Okay, so that's the question. That's the information of the question. So then here are the questions. What are the parents' genotypes? So then you go back to the question and say, okay, so here's my clue. Heterozygous for straight hair. So then we can go back to the last slide, right? I have a little cheat sheet here. So I wrote on my notes. So I have my key. So heterozygous means there's going to be a dominant and a recessive. Okay, so let's write that. So what are the parents' genotypes? So the mom is heterozygous for straight hair. That's the information I'm getting out of the question. And then it says it mates with a man who has curly hair. Well, it doesn't tell me homozygous or heterozygous. How would I know what the genotype is? Well, you go back to your key and look at which phenotype gave you curly, or which genotype gave you curly hair. And that was homozygous recessive, because straight was dominant, right? So here are the parental genotypes. Okay, so then our next step, if we go back to the instruction list, it said set up your Punnett square. So here I put in a square. So we are going to put all of the possible gamete combinations along the top and the side. So here the mom could donate either the dominant allele or the recessive allele. The dad only has a recessive allele to donate. Okay, so that's setting up the Punnett square, and then we have fill in the boxes. Right? So usually when you're filling in your boxes, the dominant allele tends to go first. It's just kind of convention, right? So instead of going little h, big H in this bottom square, I'm just going to go big H, and little h, little h. All right, so those are the possible outcomes of this particular mating. So then the second question, what are the genotypes of their potential offspring? We just wrote it in, we just filled it in, so that answers that question automatically. And then what are the phenotypes of their potential offspring? So then that's where the key comes back in. You're like, okay, so I have my offspring could be either heterozygous or homozygous recessive. Okay. So if you remember, heterozygous was straight hair and homozygous recessive was curly hair. And sometimes you might be asked for proportions or what's the ratios. So if we look at our box, we have half of the box is heterozygous. So we could say there's a 50% chance that their offspring are going to have straight hair from that mating, and there's a 50% chance that their offspring could have curly hair. So in every kid that they had with this particular genetic information, these two parents, they would have a 50-50 chance at every child of getting either curly hair or straight hair. Okay? So that would be how you could work through a genetics problem using the Punnett square and all of this new vocabulary: heterozygous, homozygous, dominant, recessive, phenotype, genotype. Okay, so some practical applications. So let's say you have a mystery. Because we have, usually with this dominant recessive, we have a phenotype that could have two possible genotypes. Can you think about what that might be? Well, the purple flower kind of is our clue because it's highlighted. So because remember, homozygous dominant and heterozygous both have the same phenotype. So what if it was a mystery? What if you didn't know what the phenotype of your purple flower? How could you figure it out? Well, we know all white flowers have to be homozygous recessive. That's the only genotype that they could have. So what they do, and this is a real life thing, is we do what's called a test cross. You know, in like lower animals and plants, obviously you can't do a test cross with humans, that's unethical. But with plants, you could do a test cross. So take your purple plant that you do not know the genotype, you know the phenotype, but you don't know its genetic allele combination, and mate that with a white plant for the flower color, because we know that's homozygous recessive. So I'm going to set up my Punnett squares. So let's have the eggs that saying the female is going to be the homozygous recessive. So we'll put that down on the side. So the egg in both of these potential possibilities are going to be the only alleles that they can donate is the recessive allele, right? So then we can put on the top of our Punnett square the two options, the two choices. So we could have a homozygous dominant would give us the purple, or we could have a heterozygous giving us the purple. Okay, so we've set up our two possibilities. So let's go ahead and fill in the boxes and take a look at our outcome. So we analyze the offspring and that gives us the clue. So in this first mating, where we have a homozygous dominant potential mating with our true breeding homozygous recessive, take a look at what we get. 100% of the offspring are heterozygous. Okay, so 100% of the offspring are going to be purple. If we go over here, we have our heterozygous crossing, oops, that's supposed to be a, there we go, lowercase p, and then we have a 2 lowercase p's here, homozygous recessive, the dominant with the recessive, and then recessive, recessive. Okay, so in this case, we have 50%, I'm running out of room here, so 50% homozygous recessive and a 50% heterozygous purple. A 50-50 split. So then if we look at our results, then we can confirm what the dad in this case happens to be. If 100% of the offspring, right, so this is this option here, if 100% of the offspring of this cross is purple, then that lets us know that that dad plant, the sperm pollen producing plant, was a homozygous dominant. If half of the plants, right, so here's our 50-50, so if half of the plants are purple and half of the plants are white, then that lets us know the dad carried a recessive allele. He was the purple phenotype, but had the heterozygous genotype. And with the homozygous recessive true breeding mom or the eggs, then that homozygous recessive allele came out as 50% of the offspring. Okay, so that's how we do a test cross. Okay, moving on. So Mendel was, again, looking at those 7 traits, and he also observed that these traits is what's called inherited independently. So what that means is, as flower color is inherited, it has no play on whether seed color or height or flower position or pod color or seed shape. Everything was independently inherited. So there was no connections, like if purple plants always had yellow seeds. There wasn't that connection, they were independent. And so he figured this out by actually following 2 traits. So he kind of stepped things up a little bit. Instead of following just one, he was following 2. So this is called a 2-trait cross, and it gets a little bit more complex, but hopefully you'll kind of follow in with me. We can still go through all of those rules of setting up our genetics problems. So we have to, the first step was to align symbols. And so this is what he did. So he followed seed color and seed shape to see if they truly were an independent inheritance pattern. So for seed color, yellow was dominant and green was recessive. So he assigned the Y letter for seed color. So capital Y is the dominant, lowercase y is the green recessive. Then he also tracked seed shapes. That was the smooth, kind of ball-looking seeds or the crinkly little wrinkled ones. So he assigned the letter S for this one. So S is the smooth shape, which is dominant, and capital S, and lowercase s is the wrinkled recessive trait. Okay, so we've set up our alleles. You can set up your key if you like, but we are going to take a look kind of more of how he walked through this. So he started by crossing two true breeding parents for both traits. So remember, true breeding is from generation to generation to generation. They have all of the same phenotype. So he could pretty much guarantee the genotypes are going to be either all dominant or all recessive. So they're homozygous for either dominant or recessive. So think about what would the parental genotypes and phenotypes be? So if we are taking a look at the dominant, right, so it's yellow seeds, color was dominant, and smooth seed shape was dominant with those letters, we would get something like this. So the parental genotype for the dominant would be big S, big S, big Y, big Y. So that would be the smooth yellow seeds, okay? And then we have our true breeding recessive. So this would be homozygous recessive for both traits. Little s, little y would give you green wrinkled seeds. So that's your your genotypes and your phenotypes, okay? So that was your parental generation. So he did a cross. He crossed the true breeding yellow smooth, that was the dominant, with the true breeding green wrinkled, which was recessive. So think about what happened when he first did that with the purple and white. True breeding purple, true breeding white, 100% of the offspring were purple. So it's going to be the same kind of pattern. So he crossed the parents to get the F1 generation. And so 100% of the offspring were all smooth and yellow. They were 100% of that dominant allele. But what do you think the genotypes are going to look like? Okay, so think about it. So if dad, we'll say dad is the homozygous dominant, if dad can only donate dominant alleles and mom can only donate recessive alleles, for that new diploid zygote in that F1 generation, they're going to be heterozygous for both traits. Okay, so you can see that hopefully. So the F1 generation will have the same phenotype as the dad, if you will, but if you'll notice, their genotype is now heterozygous for the seed shape and heterozygous for the seed color. Okay, so then he's going to cross that. So now we're going to get to the F2 generation. So just like we did with all the purple mating to get the 3-to-1 ratio for the flower color, we're going to see if something similar to that shows up. And we're going to set up our Punnett square, but instead of 4 square Punnett square, it has to be a 16 square Punnett square. But before we do that, we have to figure out what are we going to put on our box. So it's easy when it's just a trait, because it's just, you know, one letter. But how are you going to fill up 4 boxes when you have 2 traits? Well, let's take a look at the parents in this case. So the parents in this case are heterozygous for both traits, right? So we have a dominant and a recessive for each one. Now remember, this is our diploid parent cell. Okay, so it's got 2 copies of each allele because they have homologous chromosomes. But by the end of meiosis, when they're making their egg and sperm, they only are going to have 1 of each. So that's going to be 1 S and 1 Y. So if we take a look, what are all the potential possible combinations of those 4 letters into 2 letters? So you've got to have an S and you've got to have a Y. So let's do the dominant ones first. So that's a possibility. Right? Each parent, because both parents are going to be, the F1 is going to cross with itself. So the egg could donate a dominant for each. So dominant S and a dominant Y. Or we could have a dominant S and a lowercase y. It's a really bad lowercase y. Just if I can fix it a little bit. Okay, so that's all the possibilities for donating the dominant S. What if they don't donate the lowercase s? Well, then that can be comboed with the dominant y, right? We got any more combinations? There's actually one more. So we could do, the last combination is a lowercase s and a lowercase y. So these are the haploid gametes, okay, right? Because we start off with 4 letters, 2 copies of each, and now we have 2 letters, 1 copy of each. That's following all of what we just got done with meiosis. From 2N diploid to haploid, we go from 2 copies of every gene to 1 copy of every gene, and these are all the possible combinations. So you can see this set up in your book, and we're going to see that on the next slide. But this is on page 163, taking a look at this 2-trait cross. So let's see what actually came up. So here we have the eggs, right? So they were the F1, 100% of the F1 generation was heterozygous for both traits. That's this right here. And the sperm would have been the same way. So they self-fertilized. And so here are those 4 genotype combinations, both dominant dominant recessive, recessive dominant, recessive recessive. And you're going to see that on both the egg and the sperm. Dominant dominant, dominant recessive, recessive dominant, recessive recessive. So then just like your 4 square Punnett square, you just start filling in the boxes, and you usually keep the same letters together. You wouldn't go SY, SY. So SSYY, putting in the correct dominant recessive. So he did that. Again, we're talking like thousands and thousands of plants he had to go through and investigate their seed color and their seed shape as he was collecting his data. But what he found was this ratio, okay? So out of this 16 possible offsprings, these phenotypic representations, 9 of them, so that's these guys right here, so we have those ones, and then these 2 here. So that gives you 9 of them are the smooth yellow. So the majority of the offspring are going to be like the dominant allele. Then we had 3 smooth green. So smooth green are kind of a blend, right? So they're smooth, which is a dominant trait, but they're green, which was the recessive trait. And then we had 3 wrinkled yellow. So they're the dominant color, but the recessive shape. And then we had 1 wrinkly green, so recessive for both traits. So this is, you might see this if you're watching videos or animations, it's called the 9:3:1 ratio, also known as a dihybrid cross, or this 2-trait cross, where you cross 2 heterozygotes for both traits, you will get this particular ratio. Just like when you cross 2 heterozygotes with 1 trait, it's always going to be 3:1. So we have 3:1 with 2 heterozygotes with 1 trait, and we have a 9:3:1 for 2 heterozygotes for 2 traits. That's as complicated as we're going to get. We're not going to go for 3 traits. That would be kind of insane. All right, so with that idea that these traits assort independently, that takes us, our more modern understanding, back to meiosis. So guys, if you remember, all of the homologous chromosomes lining up on the metaphase plate in meiosis 1, they could either be maternal or paternal on either side of that metaphase plate. And so that's what this is showing you right here, is you have the different combinations of these alleles lining up on the metaphase plate. So you could be like this one, or you could be, so you have dominant Ys here, you could have recessive Ys over here, or then you could flip-flop. So there is no rhyme or reason, it's random. And so when you have these genes on these different loci on different chromosomes, they're going to independently assort. And so this is where we get, if you wanted a more pictorial representation of where we got those genotypes, the gametes in that last dihybrid cross or the 2-trait cross, this picture shows you how we end up with the, big S, big Y, little s, little y, big S, little s, big Y. So Mendel predicted this, even though he didn't know anything about chromosomes or meiosis or genes or traits or alleles. So that's pretty fascinating. He just did it with a big garden and lots of pea plants and a lot of time, because it took, the whole growing season to get things going where they needed to do. Okay, so what did we learn from Mendel? So we learned kind of what we call classical inheritance patterns. So our simple inheritance patterns. So genes are governed by alleles found at a single locus on your homologous chromosomes. There's often 2 forms of the gene. These are your alleles, and one will dominate over the other. So that's the dominant and recessive. But this is not always the case. So that's why Mendel was so lucky that he picked the garden pea, because there's a lot of other examples that we're going to be seeing in these next slides, that if he would have selected these organisms, it probably would not have come out the way it did. So luckily, pea plants do follow this basic dominant recessive, 2 alleles per gene, and that allowed Mendel to get his results. Unfortunately, well, I don't know if it's unfortunately, but his work was not really recognized until the early 1900s. So this was done in like 1888-ish, I want to say mid 1800s, but people didn't really start recognizing it until maybe like the 1920s. So 40 years went by or so between when Mendel published his work and people actually started seeing that, wow, this is important, because they're learning more and more about the cell as microscopy got better, and they were elucidating the processes of mitosis and meiosis. Okay, so let's take a look at some patterns of inheritance that do not follow Mendel's simplistic model. So sometimes you have one allele that does not dominate over the other one. This is called incomplete dominance. And so usually what you end up with is a phenotype that's somewhere in between the dominant and the recessive phenotype. So in this case, you have these horses example. So you have a heterozygote female palomino and a heterozygote male palomino. And how do I know it's heterozygote when they're both capital letters? Because there's no one dominant and no one recessive. So they just use these subscript 1s and 2s. So because you have a 1 and a 2, they're different. You have one allele and the second allele. So if we have different alleles, it still follows the term heterozygote. So if you have two heterozygotes cross, here you're going to get a chestnut horse, which is a homozygous C1. We can't say homozygous dominant because there's no dominance here. If you get two of the C2s, it's going to be homozygous 2, which is going to give you this cremello. I'm not super familiar with horse colorings, but hopefully that's how you say it. And then you'll get 50% of your population or your offspring will be like their parents, heterozygote. So it is this in-between phenotype. So you have the darker color and the lighter color, and then you have this blended color in the middle. Sometimes you might see that with like red flowers and white flowers give you pink flowers. That pink would be the heterozygote. The red would be the homozygote red, and the white would be the homozygote white. Okay. Another complex inheritance pattern is sometimes you have more than one allele. So I just spent this last hour or so talking about you have two alleles for every gene, two alleles for every gene. Well, sometimes you might have more alleles for every gene. So this is not always. This is one of these exceptions of Mendel's inheritance patterns. So for example, here we have fur color. The dominant is black, the recessive is white, but then you have these other forms. You have actually multiple alleles. You have the black allele, you have the chinchilla, the Himalayan, and the albino allele. You still are only going to have two copies for each one, and sometimes they fall, black might be dominant over chinchilla, chinchilla might be dominant over Himalayan, Himalayan might be dominant over albino. So depending on your combination, you might have, what if you had a chinchilla and a Himalayan mate? You might end up with a CCH and a CH. Right? You're still only going to have two alleles because you can't ever be more than diploid in animals. And so if you have a chinchilla and a Himalayan, if chinchilla is dominant, it would show that chinchilla phenotype. It would just be heterozygous and carry that Himalayan lesser recessive trait. Okay, so that would be an example of multiple alleles. In this case, there's 4 options instead of just 2, but you only ever get 2. So it's going to be the combination of whatever 2 you end up with. All right, another complex inheritance pattern is codominance, where you don't have one dominant and one recessive. And so here I found one just straight codominance is with chicken feather color. So you might have black and white both when they are expressed. When they are found in your genotype, both will be expressed. So if you have a homozygous dominant black chicken made with a homozygous dominant, I guess it's codominance, homozygous black and a homozygous white chicken, you're going to get your 100% heterozygous. And because there is no dominance or recessive, they're going to have both black feathers and white feathers. And so this phenotype they're calling is checkered because they have a little bit of both. We have a similar type of arrangement with our human blood groups, the A-B-O blood typing. A and B are codominant. So if you have just the A allele, so maybe your genotype is AA, you're only going to show the A proteins on the surface of your red blood cell. That's your phenotype. If you are BB, you will only show B surface proteins. However, if you are AB, since these are codominant, you'll have both. You'll have A surface proteins and B protein surface proteins. The blood types do have a third allele. So this is actually also an example of multiple alleles. So O is recessive to A and B. So if you're going to type O, I'm type O blood. So my genotype has to be OO. Okay? But you could have A blood type because A dominates over O. So you could have A phenotype with an heterozygote AO genotype, and same with B. So B blood type could either be homozygous dominant genotype or heterozygous genotype. So blood grouping is actually a multiple example of codominance and multiple alleles, since we've got that third O allele in there. All right, the next complex pattern is called pleiotropy. So pleiotropy is you might have one gene at one locus affecting multiple phenotypic outcomes. So what Mendel saw was one gene coded for one trait. So one gene coded for flower color, one gene coded for seed color, one gene coded for plant height. Well, pleiotropy could be one gene codes for multiple different traits. Like we can see in the picture, you have your single gene here, but it has effects on 3 phenotypic outcomes. So an example of this would be what's called the SRY gene in males on the Y chromosome. So that one gene on the Y chromosome has multiple phenotypic effects for your gonads, your prostate development, your penis and scrotum development, seminal vesicles, multiple parts of the structure of the male reproductive tract. Okay, another one is called polygenic inheritance. So just like the name implies, poly meaning many, genic meaning genes. So a lot of what we see in our phenotypic appearances are regulated by more than one gene. There's a few, like the attached earlobes, the widow's peak, the taco tongue, whether you have freckles on your face, whether you have, so some of you, here's a trait that's kind of fun. If you have hair on this knuckle part of your finger, everybody's got hair here, but if you have hair here, that is coded for by a single gene, okay? That would be monogenic inheritance. Polygenic, this is like skin color, hair color, eye color, height, anything that has like a wide range of traits that you might see, that's usually going to be coded for by many genes. So in this case, we have 3 genes code for skin color. And you have your 2 alleles, right? So here's your 3 genes, 1, 2, 3 for the eggs. And then you have 2 alleles. You have the light allele and you have the dark allele, okay? So let's say your eggs are donating their combinations, either heterozygotes, right? So they have one dark and one light, and same with the sperm. So then your skin color is going to be the sum of all of those alleles that you have. So if you have all 6 alleles, or the light version, that puts you way up here in this big box. So this is kind of like a huge Punnett square. So that might give you more of skin tone like this girl down here. If all 6 of your alleles are the dark, then that might give you the skin tone of this girl over here. And then everywhere in between, you have this combination of, you have 1 and 5, 2 and 4, 3 and 3, 4 and 2, 5 and 1. So those are going to be your combinations for everybody else in between, right? So that's our skin color is on this gradation, and that's going to be a clue that it's polygenic inheritance. Okay, and then I believe our last complex pattern of inheritance would be more of an environmental. It's actually not generational. Sometimes the environment itself can influence how a gene is expressed. So the Himalayan rabbit is a good example of this. Showing you how when these rabbits are in cold environments, their more exposed surfaces like their nose and their ears and their paws and their tail actually express black fur, whereas if it's in warm temperatures, it's white all over. So it's not a parental thing, it is an environmental thing, which is pretty cool. Probably my guess would be in the winter, things are really cold, and so the black fur is going to absorb the sun more and keep those colder body parts warmer when you really don't have much ambient heat, but the sun radiation can warm those more delicate parts. Okay, so the next topic is not quite an inheritance pattern, but it does play a role in how genes are passed on to generations. So again, another lucky thing with Mendel is all of those traits that he was looking at with his pea plants, they were probably found on different chromosomes or maybe far apart on different chromosomes. Because what happens if you have 2 genes that are very close to each other is they don't independently assort. They stay close together. So we call these linked genes, right? So here's our term. Characteristics whose genes tend to assort together are said to be linked. So if here you have, say, chromosome number 2, gene 1 and gene 2 are really close together, the likelihood that both of those are going to be carried into that same gamete is really high. If they're on different chromosomes, right, we have chromosome 2 and chromosome 5, say, they're totally, of course they're going to independently assort. It's going to be that random assortment at metaphase 1. Also, you might have, say, chromosome 2, or say this will be chromosome 3, and you have gene 1 and gene 2, but they're really far apart. So what event, think about back in meiosis, what event could potentially cause these genes to be randomly distributed to their offspring? I'll give you a second to think about it. So when genes are far apart, they also are not considered linked. The thing that happens in prophase 1 of meiosis, you guys remember, is crossing over, right? What if we get a crossing over, that sharing of the chromatid bits? This gene on chromosome, this gene 2 on this one chromosome could be split apart from its other gene 1 on the same chromosome. So the further they are away from each other on the chromosome, they're not considered linked. But come back over here. So I'll erase my little circle. So what if we are going to do some crossing over? It is more likely that the crossing over might happen here, or maybe down here, but it is less likely that that crossing over event happens in between two genes that are really close together. So the likelihood is those two genes will be inherited together. Now, sometimes crossing over can split them up and you get these recombinant organisms, but for the most part, the closer they are together on the chromosome, they are considered linked genes and they will be inherited together. If they're on different chromosomes or further apart on the chromosome, they are not linked and they will be inherited independently. Okay, another linking, if you will, it's not quite the same as linked genes by proximity. These are called sex-linked, and all that means is the gene happens to be found on a sex chromosome, the X or the Y. So genes that are found in the X will always go to your offspring. Both offspring. Genes that are found on the Y will only go to the male. So any genes that are located on these sex chromosomes will be donated to the offspring, which may or may not have an effect on their gender or their biological sex or all of those other kind of traits that these sex chromosomes might carry with them. The X chromosome is very large in comparison to the Y. They're not true homologs, right? We talked about that back in meiosis. The X carries about 1,000 genes. The Y carries only about 80-ish, about 78 genes. They have very few genes in common, so they're not really going to do crossover. They don't do the recombination event in prophase 1. But if you take a look, remember, XX are females, XY are males. So if there's a gene on the X chromosome that might be recessive, males only need one copy of that to express that gene. Because earlier we've been talking about if you're going to have the recessive trait, homozygous recessive, you're going to have that phenotype. If you're heterozygous, you're going to have the dominant phenotype. But for males, you only have one copy of that X chromosome. So whatever allele is on the X, that's the phenotype that you're going to express. All right, so this is what's a lot of times traits that are found in the X chromosome are more commonly found in men and boys because they don't have a dominant allele that could be found on the X that girls may have, females may have, because they have two X's. So one of these examples is color blindness. So here we have, this is just a generic, right here, this is just a generic trait. So you might have an unaffected father. So his X chromosome has a dominant allele on it. The mom has, she's heterozygous. So she's got a dominant and a recessive, but she shows the dominant phenotype because she's heterozygous. So if you're to do a Punnett square, which I'm going to do in a second, you'll see how these traits can be passed on to their offspring. The way that you do a sex-linked cross is you set up your XY cross, right? So female, male. Right? So this is your biological sex chromosome cross. So you're going to do 50% of your offspring can be males, 50% of your offspring can be females. That's just the distribution of your sex chromosomes. But what you can do is you can start assigning traits for this. So we'll do color blindness. So let's say mom is the carrier. So she's going to have heterozygote for, we'll say E for I. All right, so there's her genotype. She's female, that's why there's two X's. She's got a dominant and then a recessive. The dad, we'll kind of track this over here, he's normal. So he has to have the dominant allele on his X chromosome and then nothing goes on the Y because this is a gene that is only found on the X chromosome. So we don't even have to label anything with the Y. So then we do our cross, right? So here's our XY cross. So the top row is going to be our daughters. The bottom row will be our sons. All right, so that's how we get a 50-50 chance that every time you have an offspring, 50% chance it's male, 50% chance it's female. So now we can start assigning those alleles. So this daughter, potential daughter, could get dominant from mom, dominant from dad. They're going to have normal vision. So that's going to be this gal right here. Over here, kind of going across the way, we have dominant from dad, and then the mom donates her recessive. But the daughter is normal vision because she's heterozygous. So that's going to be this gal right here. She's a carrier now, so she has that recessive that can then be passed on to her offspring potentially. So then here we have our sons. So the mom gives the dominant allele. Y is the Y. There's no trait on that. And then so that's going to be this son over here, unaffected son, because the mom gave her dominant allele. And then there's a 25% chance, or 50% of her sons will be the recessive. And so that's going to be this boy here. Because he got the Y from the dad, that's what makes him male. And then he got the recessive allele from the mom. So that makes him colorblind because he doesn't have a dominant to overwrite it like the heterozygous girl does. Okay, and then another tool that researchers and genetic counselors can do is what's called a pedigree. So where Punnett squares are a lot of times you're predicting ahead, if here are the parental genotypes, what are my offspring genotypes? What are the possibilities? Well, pedigrees allow you to maybe go backwards in time or look at generational characteristics. And so it's just kind of a map of when you have somebody who has a trait, their shapes are usually filled in. Okay? If they don't show the trait, their shapes are empty. If you know it's a heterozygous, so you know the organism has one of each or is a carrier, then you can do the half shaded box because you know one's dominant, one's recessive. Or sometimes you just don't know. So if you're trying to do family trees back in the past, you may not know some of that information. So for example, this first pedigree on the top is a dominant trait. So if it's a dominant trait, the people who have the shaded boxes are going to show that particular trait. So kind of an interesting dominant trait is polydactyly. So 6 fingers is the dominant allele. 5 fingers is the recessive allele, which is kind of weird because almost everybody has 5 fingers. So let's say if you have 5 fingers, we'll do little f's. I'm not going to fill in the whole pedigree. That would just take forever. But everybody who's got a open box or open shape, the circles are females, the squares are males, they're going to be homozygous recessive. And in the shaded box, we know they have 6 fingers, but could we figure out what their genotypes are? They could be homozygous dominant or heterozygous, will both show 6 fingers. So if we take a look, if this 6-fingered man married a 5-fingered woman and they had 2 kids that had 6 fingers, that lets us know, and they also have kids that have 5 fingers, that lets us know that these kids with 5 fingers are homozygous recessive. So if mom only gives one of those little f's, dad has to give the other little f. So it's kind of like solving a mystery, is dad has to be heterozygous because that's the only way they could have kids that are also homozygous recessive like mom. And so that means these kids have to be heterozygous because mom can only give the recessive. So hopefully that makes sense. I won't be asking you to do any pedigree solving, but just know kind of this is a tool that we can use to analyze family traits passed on through generations. I think your book has a very big one of kind of, what is it? Hemophilia among royal family. And this can be a problem when families tend to interbreed, kind of incest or inbreeding, especially in royal families when people thought royals could not commingle with peasants, if you will. And so a lot of times these recessive traits were very prominent in these families. Hemophilia, which is a lack of the ability to blood clot, and some other disorders were pretty prominent in some of those families. Okay, so the last couple topics we're going to take a look at are recessive and dominant genetic disorders. So in human genetics, recessive disorders, you have to be homozygous recessive to be able to have that particular condition. There are some that are really bad for you, like sickle cell. We'll talk about that. Muscular dystrophy is a homozygous recessive. Cystic fibrosis is homozygous recessive. Some of them are not necessarily like life-threatening, like albinism is a homozygous recessive disorder. You just lack the ability to produce pigments. You have to be more careful around the sun, and you might have to take some vitamin D supplements, but it's not life-threatening like cystic fibrosis or muscular dystrophy would be. So where do these recessive alleles come from? Because if a lot of them are detrimental, how do they get passed on in generations, and how come they're not lost when people who have them usually die before reproductive age? A lot of these just come by random mutations. So here's your gene, there's a mistake, you get a random mutation, and it turns into this recessive form that affects the protein being made, and then you get that disorder. So it could be random, it doesn't always have to be inherited from parents or grandparents. When you are heterozygous for this particular trait, you are said to be carriers. That increases the likelihood that your offspring could have it if you mate with somebody else who is a carrier. So you could both be normal. Think of that, the heterozygous, two heterozygotes, you get a 25% chance that your offsprings could be homozygous recessive. All right, so the example we're going to take a look at here is sickle cell anemia, which is affects the hemoglobin, which is a protein that's found in your red blood cells. It binds to the oxygen that you breathe in, takes it to your tissues, releases the oxygen so you can do all that cellular respiration stuff we talked about a couple chapters ago. Well, it is a recessive trait. So let's say you have two individuals that might be normal blood type, but are heterozygote carriers. So we'll use S for sickle. So let's say here's our homozygous parents. They're normal with their red blood cells, which kind of look like this over here, that kind of almost a donut-y biconcave disc shape. So if two heterozygotes mate, you're going to get that 3-to-1 ratio. So 75% of their offspring will have normal hemoglobin, but 25% chance, I don't want to say 75% of their offspring, there's a 75% chance of their offspring being normal, 25% chance of them having sickle cell, but 50% chance of their offspring could be carriers, and keeping that phenotype and genotype in the population. So what does sickle cell do to your hemoglobin? Well, I think we saw this back in our genetics, in the proteins. When your hemoglobin is normal and then it has a mutation in it, it's going to cause that to fold a little bit differently. And so inside the red blood cell, what happens is your blood cells now take on this sickle shape. And we can kind of see a couple of these here in this slide. They're no longer round, they're kind of pokey. They get stuck in your capillaries and it makes it really hard to oxygenate all of your tissues, and it can be fatal. It's very painful also. Okay. So those are recessive disorders. You have to have a homozygous recessive to express that particular disease phenotype. There are also some diseases that are dominant, like polydactyly was dominant, but I wouldn't consider it a disorder. It's just a phenotypic expression. But there are some serious disorders like Huntington's disease, Marfan syndrome, that are caused by dominant allele. So you just have to have one dominant allele, so homozygous dominant or heterozygous, and you have that particular condition. So this picture here is of what happens to people's brains during Huntington's, which is a degenerative neurological condition. After about around 50 years old is when you start seeing symptoms, is you just start losing your cognitive abilities because your brain is kind of being destroyed. It's degenerating. So let's say we had, get my pen here. So maybe we had a heterozygote mate with a homozygote recessive. So the homozygous recessive people would be normal. So that would be the normal phenotype. They do not have Huntington's disease. And let's say the dad here does. So he's heterozygous. But here's the sad part about Huntington's disease is because the symptoms don't show up until usually after reproductive age, the traits have already been passed on to their offspring. So there is a 50% chance that that dad who starts seeing signs and symptoms after he's already had kids, there's a 50% chance that his kids are going to have Huntington's disease. So some people who know that these types of things run in their family because they've done pedigrees can get some genetic testing done, knowing what their genotypes are to determine whether they want to have kids or not, or if they do marry somebody, have their mate tested, just to make sure that there's not that likelihood. Some people want to know, some people don't want to know. That's a personal choice. Okay, so that wraps up our lecture on inheritance. I found kind of a comic here. So we have genetics. This is how it works. Not so sure about that. So I'm going to add in, this is not how it works. A horizontal stripes and a vertical stripes does not give you a checkered offspring. Doesn't work that way, but it's kind of cute. All right, so that wraps up kind of meiosis and inheritance kind of go together. We had the molecular mechanisms and then the larger scale outward appearance of what happens as these traits get passed on from generation to generation. All right, thanks so much. I will see you next time.